Hello, my name is Liesa Pomeroy, and welcome to my presentation on the connections between gut bacteria and sleep for Psych 410 at Portland State University. I'm interested in this topic because I'm a biology major with a psychology minor. My biggest interest in biology is microbiology, so I wanted to try to find connections between my studies. I have also studied the gut-brain axis previously about its connections to autism and mood disorders like depression and anxiety. This topic is important because it can offer new and innovative treatments for sleep disorders. These could be in the form of probiotics or fecal microbiota transplant. These treatment options might be cheaper and more readily available than current medications. It also offers the opportunity for greater understanding of the important topic of sleep. We spend at least 1/3 of our lives sleeping, and we do not fully understand the mechanisms of it. Researching this topic gives us a better look into how sleep might work. The first pathway we're going to look at is the cytokine pathway. The first step is the bacteria cell wall constituents, murine peptide and lipopolysaccharide. The immune cells of the body recognize these constituents and create TNF, or tumor necrosis factor, and IL-1, which are cytokines. TNF inhibits slow wave sleep, also called non-REM sleep, and induces the production of IL-1. IL-1 enhances slow wave sleep and inhibits REM sleep in rabbits. However, in rats, IL-1 has shown to have more complex effects. This could mean that it also has complex effects in humans, but this is unclear at this time. Next, GH, or growth hormone, production is increased by the cytokines. Growth hormone induces slow wave sleep and enhances it as well. The final step in the pathway is nitric oxide production, which is increased by growth hormone. Nitric oxide substances, when given to rats, increase prolonged sleep. Inhibition of nitric oxide inhibits normal sleep. This is only one pathway that bacteria may affect sleep by. The next pathway is the serotonin pathway. Spore-forming microbes elevate the TPH1 enzyme expression. TPH1 is the rate-limiting enzyme in serotonin synthesis. Once the TPH1 enzyme abundance is high enough, enterochromaffin cells produce serotonin using the enzyme and the enzyme substrate tryptophan. Germ-free mice, which are mice born without a gut microbiome, have lower levels of serotonin and TPH1. This makes sense because no bacteria equals less TPH expression, and lower TPH equals decreased serotonin. Germ-free mice also have increased levels of tryptophan. This is because of other interactions that are occurring. Some gut bacteria can use the tryptophan to produce indole. Some can produce tryptophan themselves, and others can produce serotonin using tryptophan. So germ-free mice have increased levels of tryptophan because there is no bacteria to use it in these interactions. Murine peptide, one of the bacterial cell wall constituents, can also interact with serotonin receptors. It is a serotonin-like neurotransmitter. To understand how the serotonin pathway affects sleep, we must look at the final product, serotonin. In the beginning of sleep research, serotonin was thought to be the main neurotransmitter involved in sleep. This has since been proven incorrect. Sleep is a complex process that involves many different neurotransmitters and hormones. Serotonin does have a complex effect on sleep, however. It is at its peak abundance during waking periods, lower during slow wave sleep, and at its minimum during REM sleep. Serotonin increases the hormone prolactin, which is secreted during sleep and seems to be involved in REM sleep generation. Different serotonin receptors have different effects. The serotonin 2 receptors seem to lead to waking periods. The serotonin IA receptors help to facilitate sleep and/or sleep characteristics. When given serotonin, rats had an increased arousal/waking period followed by a sleepiness or drowsiness period. So it seems that serotonin 2 receptors have the faster effect, and then the IA receptor takes effect later. In addition, SSRIs, which are a common medication for mood disorders like depression and anxiety, have been shown to decrease REM sleep. These are just the basics of the effects that serotonin has on sleep, and therefore bacteria, because bacteria helps to produce serotonin. Bacteria is symbiotic within us. It does not just have an effect on sleep, sleep affects it just the same. Sleep disorders and sleep deprivation have been shown to change the microbiome diversity of the gut. A study was done where they cut off oxygen of rats during sleep for a short amount of time to mimic sleep apnea. A change in microbial diversity was seen. Bacteria who liked low oxygen levels thrived, whereas other bacteria died off or decreased. This shows that the effects could be a cycle of bacteria affecting sleep, and in return, sleep affecting bacteria. In connections to bacteria, I came across a hypothesis for the function of sleep. It is that sleep helps to rid the cerebral fluid of the brain of murine peptides and endotoxins from bacteria. The blood-brain barrier is not perfect and sometimes accidentally allows substances or molecules in that it shouldn't. This hypothesis states that sleep is to fix those accidents. To conclude, there does seem to be some sort of connection between gut bacteria and sleep. This could be new treatment options for people who suffer from sleep disorders. However, further research needs to be done, especially human testing. We know a lot about the effects in small animals, but we don't know if the effects would be the same in humans. There are still a number of questions that need answers. For example, does each product in every step of the pathway affect sleep, or is it just the final product? How much of an effect are the products specifically from bacteria having? And would diet affect sleep via the gut bacteria? These are the references used for my presentation, and thank you so much for watching.